Hello class 12 science. Yesterday we have just started this topic deuterium ion, and I have explained about it once again, just a revision for you all. Deuterium ion are the ions of amino acids, we can say. They are colorless and crystalline solids. These are water soluble and they have got high melting points, and they behave like salt rather than simple amines or carboxylic acid. Now this behavior, the formation of salt, is due to the presence of both acidic and basic group. So in case of amino acid, we have got carboxylic acid as well as amino group. Now since this is carboxylic acid and it behaves as an acid, it can easily remove this H plus ion, and since it is a base, it can accept that H plus. So what will happen? H from acid will go to the base. So while doing this thing, it will form, this will be a negative ion and this will be positive ion, and because of these two charges generated on the amino acid, okay, so the carboxyl group can lose a proton and amino group can accept a proton, giving rise to dipolar ion. So dipolar means we have got two ions, so this is known as deuterium ion. This is neutral since it contains both positive and negative charge. In deuterium ionic form, amino acids show amphoteric behavior as they react with both acid and base. Fine. Now there is a important term, yesterday we did not discuss about it, isoelectric point. Let us see what does it mean. So when an amino acid is added in acidic solution, this I have written the deuterium form. When we add acid, what will happen? This H plus will link with this one and this will be neutralized and it forms a positive ion. So it forms a positive ion. Whereas when an amino acid is added to basic solution, what will happen? This NH3, from NH3 will take H plus and it will form water molecule, and this will remain a negative ion. So it will form a negative ion. So remember that an amino acid in acidic solution becomes positive ion and an amino acid in basic solution becomes negative ion. Now this in acidic solution, the amino acids migrate towards the oppositely charged cathode. In acidic solution, what happened? We have formed positive ion. So positive ion will move to the oppositely charged negative electrode, that is cathode. So this will move to the cathode, whereas this negative ion will move to the anode. So in basic solution, the amino acids migrate towards anode. Okay, now at a certain pH, the amino acid molecule exists as a neutral dipolar ion and therefore does not migrate to the either electrode. So at certain pH, the amino acid does not migrate either towards the anode or towards the cathode. That value we call it as a isoelectric point. So the definition tells us the pH at which there is no net migration of amino acid molecules towards any electrode is called isoelectric point of the amino acid. So the pH at which there is no migration of amino acid either towards the anode or towards the cathode, we call it as isoelectric point. Next, structure of proteins. Yesterday we discussed about it. That means how proteins are formed. Proteins are the polymers of amino acids. That means amino acids are connected with each other, and they are connected with each other by a bond known as peptide bond, or we call peptide linkage. Similarly, as we have seen in case of saccharides, they are linked to each other through a glycosidyl linkage. In case of protein, they are linked with each other by a peptide bond or peptide linkage. Now this peptide bond is formed between a carboxylic acid and NH2 group. Say for example, we'll take an example. We have taken, this is the glycine and this is alanine. Okay, so glycine, we have got carboxylic acid and here we have got NH2 group. So what will happen? From this OH and from this H will be removed as water molecule and it will form CONH. This is a peptide bond, or we can call peptide linkage. So in this way, the two amino acids are linked with each other by a peptide bond, and this combinations is known as, since it is glycine and alanine, so it will be called a glycyl alanine, and in 3-letter representation we can write it as GLYALA. So, and since in this we have got two amino acids, the peptide that is formed will be called as dipeptide. You can call it as dipeptide. So for example, when carboxyl group of glycine combines with amino group of alanine, we get a dipeptide. Glycyl alanine, if we have got 3 amino acids, then will be called as tripeptide. Similarly, if you have got 4, 5 or 6, they are called as tetrapeptide, pentapeptide and hexapeptide respectively. And nearly, if you have got more than that, they are called as polypeptides. And to form a single protein, the amino acids that are linked will be around 10,000. Okay, so that is the structure of protein, how they are made and how they are formed. Next, proteins classification. Proteins can be classified in 2 types on the basis of their molecular shape. One is fibrous protein and other is globular protein. Fibrous protein means when the polypeptide chain run parallel and held together by hydrogen and disulfide bond, then fiber-like structure is formed. Such proteins are generally insoluble in water, and examples of fibrous protein are keratin, that are present in hair, wool and silk, and myosin, which is present in muscles. So these are the examples of fibrous protein which are present in our body. Next, globular proteins. This structure results when the chains of polypeptides coil around to give a spherical shape. These are usually soluble in water. Insoluble and albumins are the common examples of globular proteins. So insulin and albumins are the common example of globular proteins. Structure and shape of protein can be studied at 4 different levels: primary, secondary, tertiary and quaternary, each level being more complex than the previous one. Okay, so we came to know that the proteins can be classified on the basis of the molecular shape into fibrous and globular protein, and we have seen the structure and we have seen the example as well. Then structure and the shape can be studied at different level, which has primary structure, secondary structure, tertiary and quaternary structure. So this is about the classification of proteins. Next topic, we have got denaturation of protein. Now let us try to understand what does this term means. Protein found in biological system with a unique 3-dimensional structure and biological activity is called a native protein. So protein that is found in biological system with a 3D structure, unique 3D and biological activity, we call it a native protein. When a protein in its native form is subjected to physical, so now this native protein that we can call a natural protein is subjected to physical change like change in temperature or chemical change like change in pH, the hydrogen bonds are disturbed. So what happens when a protein is subjected to or is made to undergo a change by changing its temperature or changing in pH, the hydrogen bonds get disturbed. Due to this, globules unfold and helix got uncoiled and protein loses biological activity. At this time, what will happen? The protein loses its biological activity. This is called denaturation of protein. During denaturation, 2 and 3 structures are destroyed but primary structures remain intact. So the example of denaturation of protein is the coagulation of egg whites on boiling is a common example of denaturation. Say for example, when you boil egg, what will happen? The liquid portion will get coagulated. That is a common example of denaturation of protein. Another example is curdling of milk, which is used due to the formation of lactic acid by the bacteria present in milk. So the second example is the conversion of milk into curd, which is caused by the formation of lactic acid that is present in the milk. Today's assignment number 7. Question number 1, what is deuterium ion? Give an example. Question number 2, define isoelectric point. Question number 3, what is peptide bond? Question number 4, define dipeptide. Give one example of formation of dipeptide. Question number 5, give 2 examples of each of the following: fibrous protein and globular protein. And number 6, what is denaturation of protein? Complete this assignment and send it in my mail.